Good afternoon, everybody. I am very excited today to introduce Dr. Socrates Trujillo as our speaker for today's Educators Call. Um, I hope everybody's having a really great end of summer. Or I refuse to acknowledge it's getting to be the end of the summer. So I hope everybody's having a great August at least. Today we are here with Dr. Socrates Trujillo to talk a little bit about updates from the FDA Office of Applied Microbiology and Technology, or OAMT, and a couple of notes before we get started. Just as a reminder, this is our normal screen about the Educators Call group. All participants are muted. There will be time for questions and discussion at the end of the meeting. We're going to have some time at the end of Dr. Trujillo's talk today. As you think of questions, feel free to use the chat box or Q&A. We will likely hold most of the questions until the end, but feel free to enter things as you have them, and I will keep track to make sure to queue them up for Dr. Trujillo at the end. This session is being recorded, and the presentation will be shared via the listserv and on our website after the call. And just as a reminder, the views and opinions expressed in this presentation are those of the speakers and do not necessarily reflect legal advice, views, or positions of the PSA and its members, nor any entities that they represent. So with that, I am excited, I mentioned, to introduce Dr. Trujillo. Socrates, or Dr. Trujillo, is the Director of the Food and Environmental Safety Division at the Office of Applied Microbiology and Technology, OAMT. And today his talk is really going to be talking about updates and some of the research that they are conducting at OAMT. Currently, Dr. Trujillo leads, supervises, and collaborates closely with the office director to provide recommendations to ensure that the research being conducted supports regulatory programs such as the Produce Safety Rule. And Dr. Trujillo has worked for the FDA since 2001 in various roles, including as a researcher, advisor, leader, member of various working groups. I had the pleasure to first work with Socrates. I think we both co-presented a long time ago at a GIFSAN Good Agricultural Practices training in, I think, Honduras, maybe. And then I had the pleasure of working with him a lot more during his time working with the Produce Safety Network. And then he was also part of the team that developed the FSMA Produce Safety Rule. So with that, I am going to turn it over to Socrates to share his screen.
Donna, thank you very, very much. Good morning, good afternoon, everybody. It is definitely a pleasure being here. And as I'm talking, I am going to be sharing my screen. Make sure that you guys see what I am going to be talking about. And if you do, Donna, let me know. It should be the presentation title. Again, yes, Donna, I want to say we met in Honduras. What year was that? You were so young, still are, but I think you were like finishing kindergarten, I think, when you were already teaching produce safety. I was already graduated. That's right. So it's been a pleasure working with you and your group. Thank you very much for the invitation. So for everybody who knows, I've been working for the agency, as Donna indicated, since 2001. And strangely enough, produce safety has been one way or another one of the activities that I started doing during the years of me joining the agency. My areas of expertise was mycotoxins. So again, produce safety, different area. Again, now the definitions of produce has changed to what we used to go into what is related for the agency, and the produce safety rule has more limited information of what we are going to be calling produce and what type of produce safety we're going to be working on for what we call the program offices. And I'm going to be talking about that. Some of you have been interacting with, have interacted with the agency and the FDA and what you used to be called the food. Oh my God, the Center for Food Safety and Applied Nutrition. It's only been not even a year, and I don't remember the acronym. Welcome to our world. So CFSEN no longer exists. We were absorbed what we call now the program, a program office, or a program within FDA, and that is a human foods program. A lot of components from different parts of the agency migrated into the human food program. And the advantage that we have now is that all the laboratory work that was done at the agency level has to do with food regulated by the actual agency what migrated into what we call the OLOAS, the Office of Laboratory Operations, right? So that's where we are, and I'm gonna be talking about all that, and again, you please more than welcome to type some questions in the chat. Donna will let me know, and that way we will make sure that we move forward. Did I lose missing everything? Is okay? You still see the
Yeah, and
presentation?
so I'm not seeing your slides.
Yeah, that's what I thought. As soon as I saw that, it changed. I don't know why. Let me do it again.
Yeah, no worries.
Um,
I'll let you know if I
now
see them.
No, it should be share. Here we go.
There we go.
Had to again one more. I was telling Donna when I
There you
joined,
go.
there was one more click. One more click.
It's
Here we
always one more
go.
click.
So
Looks
this is
good.
again. I'm not going to start from where I said because I already spoke for 5 minutes. And all the people who knows me, you need to let me know. Hey, time's up. I'll try to be as informative and as succinct as possible. I indicated the Office of Applied Microbiology and Technology is part of the Office of Laboratory Operations and Applied Science within the Human Foods Program. This is what, and some of you remember, used to be called CIFSAN. OLOAS is a new super office that we created, in the Office of Applied Microbiology and Technology compiles every work that we do in microbiology, technology, and some chemists. But in our case here, because we're going to be talking about produce safety, and you guys are helping us with the produce safety rule, and the network is more about implementing helping us with the implementation of the regulation and education and outreach. We will concentrate on produce safety, but with the understanding that OAMT does more than just produce safety, and we'll go in that direction. Just as for of you who have had the opportunity to see our new website, you will see that this is the Human Foods Program, right? Mr. Kyle Diamantis is the Deputy Commissioner for Human Foods, an incredible, great advocate for not only produce safety, but research and other aspects of what we do. We are very ecstatic of but all the support that we're getting from Mr. Diamantis to make sure that we develop what is needed to properly implement all the regulations within the agency, in this case in food science and because of Human Food Program, but well identifying the key players to make sure that we get the support that we need to continue developing what you guys need to do the education and outreach, right? So you can see the Human Foods Program is compiled for all those offices. So we are where the little green rectangle is located at, the Office of Laboratory Operations and Applied Science. That's where we live now. Before, when the CIFSEN was the center, we had a center director, and the office director used to report directly to the center directors. Now we have a deputy commissioner, we have a super office, and the offices report to a super office director, and the super office director reports to the deputy commissioner. Now, how that has changed, we will talk about that. There's a more streamlined, surprisingly, communication and a better understanding of what we do and what you guys need to do the work that you are interested in doing regarding the produce safety rule, education, and outreach, right? Now, again, what we're talking about this, you can see now, this is the octopus that we work under. This is the Office of Lab Operations and Applied Science. I hope you guys see my pointer. This is the Office of Applied Microbiology and Technology. This is where I live, literally. And for all of you who used to somehow know about ORA, the Office of Regulatory Affairs, they used to have our labs in the United States, what we call the FDA labs. They all have migrated into what we call the Office of Regulatory Testing and Surveillance. And all these labs that you see here listed are those labs who were called RA labs. Now they're ORTS labs. Nothing changed by them. It is now the report to the Office of Regulatory Testing and Surveillance, and that director reports to OLOAS, Dr. Kiss Hale, Admiral Hale. And now we have a more direct communication between the labs, the researchers that develop the methods, that not only microbiology, but also chemistry and toxicology, And then we have an Office of Scientific Coordination and Computational Science, who actually does help us with all the work that we do for example, the genome tracker move there. You guys know about that. It's right here. And again, we had a lot of research advisors at the program level who support all the work that we do because of the experience with industry and our academia separate, a different point of view. Remember, we need to have all discussions with everybody who has different opinions to make sure that we provide everything that you guys need. That's something very challenging here with us to make sure that we open to every single opinion and listen what needs to be done to produce the data. Because remember, the agency is science-based. We do our work based on what is needed to protect public health. And the reasoning behind becoming OLOAS is to continue doing that in a more streamlined way of communication. Again, the labs who implement the methods that we develop, chemistry and and microbiology, and with the support of OSCCS, makes us OLOAS, and that's what I wanted to talk to you. Again, the Office of Applied Microbiology and Technology, right now we have an acting office director, Dr. Nathan Anderson, who actually sits in Chicago. He's our acting office director since the beginning of July, July 1st. So it is my understanding that this position will be filled hopefully soon. Anybody who's interested, please look into fda.gov for any updates on jobs at the agency. We will be actively looking for the best candidate to become the office director of the work that we do to support public health from the microbiology and technology and some chemistry work. The office is divided in 4 different divisions. As I indicated at the beginning, or Don indicated, I'm the director of the Division of Food and Environmental Safety. That is the work that we do here. Dr. Brown, Eric Brown, some of you may have worked with him because he used to be the director of the Division of Microbiology in what it used to be the Office of Regulatory Science, what it used to be CIFSEN. He migrated and became the director of the Division of Food Safety Genomics. And then we have Dr. Anderson, Nate Anderson, who is the director of the Division of Food Safety, Food Processing Science and Technology, who actually sits in Chicago at the Moffitt Center. And then we have Dr. William Burhart Bill. Dr. Burhart is the director of the Division of Seafood Science and Technology. Some of the work in produce safety is done with Dr. Anderson. Some of the work is also done with Dr. Brown. The majority of the work in produce safety happens under the Division of Food and Environmental Safety. And again, but so you guys understand, the 4 of us collaborate. We come from 3 different offices from the past. Again, we were literally dissected and to put together into the best structure that supports the work that we do at the agency in produce safety in this case. And that's where I want to concentrate my talk today. So all this was background to let you know that what we do, it is to develop methods. We have a mandate by the Congress of the United States to develop tests and sampling methodologies. And again, yes, it is to be deployed on the borders to make sure that we detect adulteration of foods, right? So that is a mandate that we have in the U.S. Code that we need to develop these methods. Of course, not everything that we consume in the United States is produced outside of the United States. So a lot of the mandate that we have is not only, of course, to do the detection of specific pathogens or contaminants on imported foods, but we utilize the same methodologies for the detection of such contaminants on FDA-regulated products that are part of what we do here in the United States, where we grow, harvest in the United States and process as well. So we don't have different methodologies. Use the same methodologies for produce or FDA-regulated products who are coming to the United States. The one is produced here in the United States, but that's a mandate. That's where we exist. And the methodologies that we develop are implemented in our ORS laboratories, but we also collaborate very closely with some states. If we have programs And we'll talk about that we actually do also train and make sure that some of our state partners are also creating the data that we need to protect public health, right? And again, um, that's not all we do, but that's our mandate, right? So in the meantime, we do method development and we implement them in our ORS laboratories to detect contaminants of FDA regulated products coming to the United States. We will continue existing, and that's what we do. Um, the interesting part is that OAMT was recently created, and the word creation really means that we were put together. We existed before, as I indicated. I was a division director for a former division in a former office that we used to do microbiology. Eric was still, he still is, but he was a division director on in an office, and he was a division of microbiology. Nate and Bill are division directors. Their names didn't change, their divisions didn't change. We all migrated and became OAMT because it's the best thing to do. It was a logical thing to do, and we were looking for the opportunity to all work together. And we created as the four division directors a research priority to support the work that we do for the agency regarding public health. Based on the mandate that I just told you about method development, one of our research priorities is exactly that, right? We need to make sure that we have the methods that are going to detect the specific contaminants, pathogens, in this case, and the matrices that we need to make sure that we identify the pathogen. And then we not only do to develop it, but we need to properly implement it. And for us to properly implement those, we need to validate them. And not only do you validate them, but we also need to continuously make sure that our laboratorians, the people who's actually implementing the methodologies on their labs, are proficient of what they're doing. So that's what we do here. We coordinate, help the coordination of the proficiency testing for all the labs that implement the FDA methodologies and make sure they're properly validated and they're following all the requirements for certification at the international level. I put in parentheses the divisions that actually do the work. And as I indicated, the Division of Environmental Food and Environmental Safety, the Division of Food Safety Genomics, Moffitt Center, and also what we call the Dolphin Island, the SST. We all do method development, we all do validation, and we do all help on the proficiency testing of all the last implemented methodologies. Then we do produce safety, as I indicated, of course. Surprisingly, all the four divisions that produce safety in one capacity or another, or some of our biologists who do produce safety in biology do live in the SST. A lot of the work that we do in produce safety happens with minimal processing, so it happens in the DFPST, and of course, functional genomics, that I'll talk about that in a minute, and environmental safety, food environmental safety, we do all produce safety. And this is very interesting. One of the reasons that the agency, decided to streamline the organization is to foster collaborations. So I think we were the poster child of collaboration. I hope you guys know that we always look for collaborators. Some of you work in academia, some of you do have friends in extension. And I hope you guys know that Dr. Brown, myself, Dr. Burhardt, and Dr. Anderson, we always collaborate with academia and industry, and that's not a different among ourselves as well. Several of our projects on produce safety involve the four divisions. I indicated, of course, the SST sits in Dolphin Island, Alabama. It is the seafood safety, so that's what they do. And our sister division in Moffitt Center do assist in some of the seafood safety work that they do because they have chemists as well. So some of the toxins on seafood safety toxins they work together. And again, we started thinking about all the projects that we have, and when we realized we work pretty close with academia and industry to inform and mitigate risks. A lot of the programs that we do is to generate the data that we require to provide some information to our partners, in this case, you, the academia, who are going to communicate that information to the industry. In this case, the industry working in produce safety, and then we we knew we were doing this. So instead of doing the others, like, everybody has says, oh, I do this and others, we decided to name it. Now it is part of our research priorities to do that work that we were always doing, but now we put it in an area to inform our leadership. We always communicated with industry, we always communicated with academia, and we provided information that they need to bring that data, that information to the industry, to the actual users of that data, and to make sure that we can improve the conditions, right? And again, um, risk zero doesn't exist, zero risk doesn't exist, but can we, move in that direction, right? And that's how we decided that our research priorities should be doing that. And again, that's where a lot of the work we do with the produce safety rule lives, right? For all these modifications, improvements, or specific developing specific guidance documents for a specific industry and specific location are captured. And of course, for some of you who not only work on the produce safety rule, but also the preventive controls, our partners in the FPST are 100% committed to work with the preventive control regulations. So we do that a lot as well. So for the last, 10 minutes, I've been talking to you about what we do. And now I'm going to give you a little bit more of the insight specifically about produce safety. And as I indicated, we have some chemists because seafood safety, as indicated, biotoxins, we need chemists to identify and characterize the presence of those contaminants in seafood. And processing controls, preventive controls requires some chemists, not only for allergens detection, but also to make sure that the processing is not altering the chemistry of the packaging or the food itself. So some of the chemists still migrate with us to continue doing the work that we're doing. Again, at OAMT, yes, Office of Applied Microbiology and Technology, and parenthesis, little parenthesis, and some chemists, right? Now, what do we do? So produce safety, right? So I was lucky enough to grow in the agency in the direction of supporting all the efforts that we did in produce safety prior to what is the produce safety rule. And I arrived to the agency in the midst of what used to be the good agricultural practices. And Donna and I, yes, I had the opportunity to work with her in Honduras when we were implementing the good agricultural practices that Gibson used to have that training. Some of you may remember with Jim Rushing, but before Jim Rushing, we used to have other leaders who actually identified key individuals who had some experience and then brought us together. And I was very lucky to attend several of those trainings. That is the predecessor, the good agricultural practices is the predecessor of the produce safety rule. And again, so I always had the opportunity to know, okay, what do we need? What questions do we have? Whether the industry can or cannot do. So when I was migrating from the produce safety network, what used to be Dr. Torrance, in, she knew I was interested on moving forward. One of the things that we always had was we need more data. Yes, the produce safety rule is based on data, but we still need more data. And one of the things that I wanted to do is like, well, can we generate more data? And I was lucky enough that I was selected to become the branch chief of a branch no longer exists, where we were implementing produce safety. And it was the idea to answer some of the basic questions to me that we had. Do the pathogens survive when we are exposed to extreme conditions where we grow in the environment? So with that idea, I knew for a fact that the agency has isolated uh, outbreak strains for several commodities that we regulate at the agency, and they live in the lab. They're still in the lab. We were not doing anything with them. They would just be sitting there because we identify them, we characterize them, and then put it into a freezer. And then I mentioned to my supervisor, Dr. Torrance, and Dr. Hyatt, she was the office director then, that if we buy some chambers and we obtain those isolates from our labs Can we see how they behave, and are we detecting them? Because that's the other beauty of that our methodologies, yes, when we develop them, they were developed with usually strains that are lab strains and or attenuated strains and or surrogates. But this was the opportunity to actually obtain and isolate from an outbreak investigation and then exposed or produced during pre-harvest and harvest conditions, and see, are we detecting the pathogens that we're detecting, and what is it migrating from the soil and or the water and surviving in the environment, and in this case agricultural environment, and how we're detecting it. So we started with what we call growing rooms, growing chambers. Here in OAMT, we had what we call the smaller ones, a very small footprint, to start growing plants and produce, and demonstrate to my supervisor that we knew what we were doing. And then we started with that. We started with six Gen 1000s who are rich in chambers. We can grow small produce, and just so we started with herbs, just to demonstrate my supervisor, hey, we can do that. And when my supervisor saw that we knew what we were doing, we started bringing the microbiologist, say, okay, this is your matrix, this is your environment. I want you to test what you know and make sure that you are detecting what you said you're detecting with the method that you developed. Why is that important, relevant? Because that's a reality we live in the environment, right? There's a lot of environmental constraints that we're starting to understand how they impact the survival of those specific pathogens. But again, all those environmental constraints affecting the identification, isolation identification of our pathogen with the method that we developed. So we started with what we call the chambers. So this one right here is my supervisor surprised me with the idea of having more than the Gen 1000 smaller machines, and she supported me to get a GR 128 that is as 128 cubic feet of space to grow. And I remember telling my technician and say, okay, I'm sorry, but if you have green thumbs, anybody can grow tomatoes, everybody can grow, the majority, some of us cannot, cilantro and all that stuff. So I was, can you grow tropicals? And to me, if you can grow papayas and to the fruit production, we're golden. And he did. We were able to grow papaya trees and do fruit production, and then we utilized those papayas to do some washers with contaminated water and see the survival of salmonella on recently harvested papayas and put in the environment, and then in storage conditions, and to make sure that we see the survival of salmonella. So we're going to be coming out with a paper about that. But again, that was sidetracked to the work that we do, but we didn't stop there, right? So I said, well, yes, we can grow them in the lab, but can we grow it outside? And it took me a while, but I convinced my then supervisor to give me a little piece of land outside here on our campus. And we are going to our fourth cycle of production of romaine lettuce in the outside that we use for, let me just mute this. I don't understand. Somebody's is pinning me and they don't understand that I am not available. So give me 1 second because it's becoming annoying to me, and it has to become annoying to me is annoying to you. There, get quiet. So again, so we were growing romaine lettuce, and the reason why we started to do romaine lettuce is because yes, we can grow produce and we can contaminate it, but how about the survival when we minerally process that produce? So again, we demonstrated to my supervisor we know what we're doing. We were able to already publish some of the work that we're doing in survival and transfer of pathogens, and again, isolated from outbreaks into a growing conditions. And she supported me to buy a washer, a produce washer here. And of course, if I go to the terminal market and I buy 100 boxes of romaine, my supervisor was going to not be happy about it. So we decided to grow the produce and then utilize that produce to feed it into the washer, because our methodology needs the identification of the pathogen in wash water, and for us to produce the wash water, we need a lot of produce, right? And again, the industry, although they like to collaborate with us, was not going to give us wash water. And our partners through the USDA gave us one time, and that was it, right? So Anyway, so now, as I say, it's going to be into the fourth cycle of growing romaine to develop the water that we need for the detection of specific pathogens of concern in wash water, and see if our methodologies are really detecting the pathogen we said they're detecting. And not only that, but it is the inhibitions there modifying. Do we need to modify our detection method because of all the inhibitors that may be present that we would not know unless we do this? And again, we do not contaminate the produce in the open environment. We do that inside of the labs. We use clean produce to bring it into clean, right? Because we run it outside, but at least we don't have the a lot of the constraints or concerns from the industry. We wash it, we obtain the water, and then we test for the presence of pathogens after we spike that water. But then we didn't stop there. And how about hydroponics, right? A lot of the people is moving in that direction. Do we really know what is the environment of hydroponics, how the pathogens survive, how they transfer, how to get in hydroponics farming community? So we now have a new laboratory who actually Dr. Machado from the University of Maine accepted a sabbatical here, and we work with him, and we already have enough data for a publication of the presence of salmonella in a in a hydroponics environment. And again, stay tuned for that. And again, we develop all this research to provide information so you can communicate with the industry about, hey, this is what we're observing. Can you guys help us with that, right? And again, but we didn't stop there, right? Because one of the biggest problems that we have, I don't know if you guys have heard about a parasite called Cyclospora cayetanensis specifically, is unique because we don't know much about it. We haven't been able to identify any other organism that replicates inside of but the human body, the intestinal tract of the human body. So we're trying to trick Cyclospora and make it believe that it's inside of the human body, but it's not. And we're using state-of-the-art technology, organ on a chip, where we actually are growing our own gastrointestinal cells and then bringing the Cyclospora inside and see if we can reproduce it. So we can do all the experiments that we need to identify more about this pathogen, right? So again, I'm very lucky that we have an incredible cadre of scientists who are not only smart, but also interested on answering the questions that you guys have to be able to provide that information to the industry. Again, why do we do research at OAMT? I indicated we do method development. It's on Monday. We must do that. But we don't stop there, right? A lot of the methods of detection that we develop will end up on identifying pathogens in produce and or other regulated FDA products. And our Office of Compliance needs to make sure that when they're going to bring a case, right, to say, hey, industry, what are you doing, right? They need our support to make sure, number one, our is the data that our laboratory is producing reliable? Are we confident that the method that we developed is detecting the pathogen we say we're detecting? And right, can we pursue a compliance activity based on the data that is being captured? And of course, we will also have the industry saying, wait, I'm using this other method. And then we evaluate those methods as well. Are those methods comparable? But again, right, a lot of the data that we have still generating has to be used to support regulations and guidance. And again, that's where you guys come in. All the observations that when you're doing your education and outreach and you notice data that is missing, that there's some parts of the regulations that you think they could be improved. If we had specific data to answer those questions, please send them my way. We work very close with the program office, the Office of Microbiology, Office of Food, Office of Microbiology, microbiological food science, and new acronyms. I'm still trying to remember them. OMFS. That's what we call the program office. They are the ones who interact with you guys. That's the ones who develop the regulation. And we work with them very closely to say, hey, what data do you need to make a specific modifications to data guidance documents, or also to make recommendations to the industry as well, right? So okay, we just generate the data. We just don't tell you what to do. Fortunately for us, OMFS is the one who does tell you, hey, this needs to be done. And majority of the time, fortunately, is based on the data that we generated here. I hope some of you have already seen this, the three-legged stool of food outbreak investigations, right? I will tell you that the majority of the time, we do not get the culprit organism associated to the outbreak and produce safety. By the time we identify what the vehicle is, the majority of the time the produce is completely out of the market, it's already consumed, produced the disease in humans, and we don't have access to it anymore. And again, it's based on the epi and the traceback. In some of the areas, it is very difficult to identify the pathogen, but by the time the epidemiology was done and by the time the traceback happens, we don't have access to the samples anymore, right? So that's what the experiments that we're doing here. Can we do something faster? Can we do something more accurate? Can we do something in a time frame that would allow us to reduce the onset of illnesses associated with this consumption of specific produce that is contaminated with a specific pathogen? But then, for all of you who have followed the development of whole genome sequencing, we're getting there where we started with the plate with a pathogen isolation now, and then we move forward to do the identification. Some of them versus serology, say salmonella. That is a, comes to mind. The majority of the time, all those names are salmonella based on serology, but also ESTEC. But then we start move forward into the genome and we start doing subtyping. I don't know if you remember what PFGE used to be, that we would have parts of the genome, and depending on how parts they were formed based on the enzymes that we used to cut them, that we form patterns. Well, we move forward from there. Now we're doing whole genome sequencing, that we can actually identify the whole genome of the organisms that have been isolated from the samples that we get. We do all that here with us, and that becomes a tool. Now the cost of do a whole genome sequencing when we started doing here at the agency has dramatically changed and been modified. So we'll talk about that in a minute. But again, I keep on going down. We have a mandate from the Congress of the United States. We must develop methods. We must validate those methods, and we must do it for detection and characterization of those pathogens of concern, right? So again, what are the pathogens of concern? Some of you guys know already salmonella, of course, is E. coli, the ESTEC and non-ESTEC toxigenic E. coli. We have listeria. We have some specific parasites and such as Cyclospora cayetanensis. We have viruses such as hepatitis A and norovirus. So that's where we concentrate the majority of the work and everything else, right? There's some emerging pathogens of concern that we are developing methods here to make sure that we try to be ahead of the curve. But as you know, because the majority of the a lot of the produce that we consume in the United States come from other countries where their environmental constraints or pressure is completely different. There may be some pathogens that we don't have in the United States that we need to be make sure that we develop method for detection to try to avoid the presence of the pathogens in our data and our daily intake of food, right? So we started with the classical plate count to now characterization, very specific morphology part of the organism, expression of the genes, proteins that they're expressing during they grow, and lately we're now doing whole genome sequence of every single isolate that we capture of an outbreak identified organism. So we have all the repository here with us, and we have thousands and thousands of whole genome sequencing that live here with us now to make sure that we understand the pathogen to the point that some of the research that we're doing is to identify why specific pathogens are surviving and constantly coming year by year with minimal modifications of the genome, what makes them survive and not being altered as much. And we can identify years of data where pathogens are being moved from one environment to the other and end up in our food supply. So why is that happening? And the only way to do it is to start asking the whole genome and start seeing specific characteristics of those organisms to see what is going on with the adaptability, why are they migrating from one location to the other, and is this something that we need to be preventive for other organisms of concern, and can this data help us to prevent those illnesses, right? So again, that is the preamble to the genome tracker, right? One Health. All of you guys know that we finally identified, finally with the last 20 years, that we are not alone. That nothing that happens in our food is isolated. That we grow and exposed of food that we're growing to completely different factors, from the water to the soil to the animals to the processing to the way we process the food, to the way we consume and store it in the house. So we are part of One Health. Everything that we do has that focus on. That's exactly what we need to make sure that we collaborate with all these different groups who allow us to understand why the organisms are moving from the natural environment to our food supply. So that's what we're doing. We are working with the international organizations, not only the United States domestically, but also international, with different groups. We have different projects that allow us to collect whole genome sequencing from different organisms and to start seeing how it migrates, how it survives, and how it produces diseases in humans. We this activity happens within the United States in collaboration with the industry, and that's something I wanted to let you know. So we have laboratories who are helping us with the sequencing of those isolates. We have our 7 FDA labs, right? And I indicated some states work with us, what we call the laboratory funding model, flexible funding model, the LFFM. That's another acronym that we use just in case we didn't have enough. And that with the states. So some of our state partners are part of this. We give them money for them to isolate and characterize at whole genome level these organisms. And then, fortunately for us, Congress realizes that a lot of the food comes from outside of the United States, and we have a specific mandate. In this case, not part of produce safety, but is how the model that we use for produce safety now is utilized for the shrimp mandate, where we actually isolated specific pathogens from shrimp produced outside of the United States and start adding that information into our database to try to understand as well how that's migrating. I know it's not produce safety, but it's part of the whole battery of data that we currently have to try to understand what can we do to reduce the likelihood of diseases in humans. And again, through food, right? This is important, you guys, to know, because everything you're doing regarding education and regarding the produce safety rule allow us um, to modify the specific guidance based on the experience that you capture when you're interacting with the industry, right? So that's very important. And yes, we talk about genome tracker. Genome tracker was developed with bacteria on mind, Listeria, Salmonella, E. coli. Now we have Campylobacter. We're doing what we call the viral tracker, because now we're adding RNA data from the viruses. And then we do have a cyclo tracker, right? Because we're not creative enough, that now captures data from Cyclospora cayetanensis, and to try to understand. Of course, it's not a whole genome sequencing because it's a huge genome for the parasites, but we're doing SNPs of the genome to try to see which one is being informative, and do we see different parasites coming into the United States or being captured outside of the United States in our food supply. Why do we do this? To try to reduce the likelihood of outbreaks and try to improve our health here in the United States, right? Again, the opens of virology and parasitology. We do a lot of work with virology and parasitology. My group here does the work in Cyclospora crypto and toxo. Again, I indicated toxoplasma because it is one of the pathogens of concern that we've seen. That it seems like it is coming through not only with felines exposure, but it seems that may have a component depending on the regions of the world where the produce is being grown and harvested, with the presence of felines in that environment. So we're trying to be ahead of the curve. We haven't seen it in the United States, but we collaborate with the partners from other parts of the world. It seems to have some signals about toxo in produce. Stay tuned for that one. In crypto, all the data that we know about parasitology started with Cryptosporidium. We thought we were ahead of the curve with crypto, but it seems to be re-emerging. So all the methodologies that we developed in the 1980s for crypto needs to be modified. So Dr. Durigan, who is our lead, one of the lead parasitologists here, is making sure that we develop those methodologies to see the presence of crypto in our world here. And again, right, we do all this in produce. We get the viruses, we get the parasites, we add them into our water or soil and see survival, and then identification and characterization on the plant environment. I've been talking to you about environmental microbiology. Without you guys noticing, all the work that we do here had that component. We're making sure that methodology that we develop for identification and characterization of all those pathogens of concern are being really well characterized, or are really giving us the data that we need, and the information that we give when we have those pathogens from the real world. So again, we work very closely with some of you guys with extension. If you work on extension for do outreach, and then we need to make sure that some of the data that we're generating here comes from the interaction with you guys as well. And again, as I indicated, I wish I could tell you we're isolated, that what we do in the farm is only that specific environment, but that's not true. We need to start questioning what's happening around the farm, where the water sources, the pressure of the animals on the one side, and for the wind as well, and how those pathogens are migrating from the wind, from those concentrated animal facilities to where the farm is. And again, we do this to the request from our other offices here. The survival of salmonella on an onion. I mean, I'm a food scientist for background, and I knew onions do have natural antimicrobial components, and it's a very hostile environment for where we thought it was pathogens, right? But now we have associated salmonella to onion consumption. So we are doing several experiments here to understand why salmonella is surviving in such a hostile environment, what had happened, if there anything in the genome who allowed the specific strains to survive, or are they interacting with plant pathogens? Is the plant pathogen giving them that opportunity to survive and thrive, or at least survive and produce diseases in humans? Again, technology, we do a lot of the work. There is a lot of the food who is minimally processed, and then we assume that the pathogens have been eliminated. Surprise, surprise, they're not. So we're making sure that we get sprouts, specialty mushrooms, peanut butter, hard-boiled eggs, all that items that we were for us minimally processed, and the pathogens are surviving. Why are they surviving? And is there something we need to learn out of that to reduce the likelihood of contamination? As I indicated, we support proficiency testing, or one of our groups here does work with around 25 proficiency testing events every year, when we actually produce around 8,000 samples that support approximately 500 labs in the United States. And then we collaborate with several partners, not only on the food program, but our sister agencies between the FDA, but outside of the FDA as well. And again, are we missing something? We don't know. But this is here, you guys. I'm here, and I'm very lucky to be invited to talk to you, because you may know something that we don't know, and we need to hear from you. We need you to tell us what we don't know, so we can start investigating and say, hey, now we're trying to trick a pathogen to believe that it's inside of the human body, right? So we can reproduce it. what else is out there that we need to start looking into it to reduce the likelihood of contamination. And we do more than just this. I'm going to be provided this presentation, Donna, for you guys to distribute it properly. It's been clear by my leadership. And again, very happy to be here. I'm actually going to be supporting an outbreak investigation soon. That's what we do too. We get deployed to other parts of the world to make sure that our partners have what they need to make the appropriate calls, either try to identify the source of a specific contamination. And my friend just told me it's time for me to get quiet. And fortunately for me, I'm almost done, and this is who we are. This is how we support our different components of the office, and then the human food program. We do everything that they ask us to do, to the point that they say, hey, can you do that? And I would say, let me ask, and the answer was going to be yes. And with that, any questions?
Thank you, Socrates. That was great. I feel free to ask any questions in the chat box or Q&A. If you want to verbally ask your question, you can either turn on your video and wave at me, or you can hit the raise hand button at the bottom of your screen, and I can unmute you. And as people think about their questions, I do have a question that I had. I was just curious. So, Socrates, you mentioned a lot of the work that you all do at OAMT. How do you determine or prioritize what research you might do in your office versus some of the collaborations or additional, I guess, opportunities for funding in working and collaborating with external folks?
Thank you, Donna. Really, really good question, because right prior to this presentation, I was talking to one of my branches for that specific reason, is that I indicated that we now, CIFSEN, now HFP, is part of the agency, and a component, sister component of the agency are officer inspections, investigations, who actually have the opportunity to go out and collect data and samples and all that stuff, right? So method development also is part of what they do, right? So we develop the method for them to capture, and it is what they need. What do they need for them to actually identify the specific produce, specific pathogen, send them to the labs, and then the labs do the identification characterization. So customer, one of our customers is our Office of Inspections Investigations. Is their priority, is how we say, okay, this is a priority for the agency. We need to make sure that we develop method to identify a specific pathogen in a specific commodity. So that's one of our components. The other components will be our Office of Microbiology, Microbiological Food Safety, OMFS, where they actually say, hey, we have this gap. We're trying to put together a guidance, but we don't have enough data. Can you guys help us? The anion outbreak that we right now have, the questions, is because OMFS came and said, we don't understand why Salmonella is surviving in onions. Can you help us with that, right? And then we have Office of Compliance and Enforcement, who says to us, there's cases, core is just cases in OCE identified with the presence of the specific pathogen in a commodity, but our labs are not getting them. Can you please do and see, can you improve our method of detection and identification, right? So that's how we do here. Yes, there's a lot of scientific curiosity, but that's not who drives us, right? Yes, we want to continue doing the work. We want to develop the data that is needed based off our customers, um, but we don't do basic science. Everything we do is applied and based on the questions that our partners have for us. And trust me, there's a list that we will never be able to do all the work, and that's when we say we can collaborate. So let's see. We don't have time. We don't have the expertise, or we don't have the tools to answer this question. But we know, right? USDA may have it. We know Cornell may have it. We know UT may have it. We know UC Davis may have those. So let's work, let's collaborate, and then answer the questions together. Because that's your beauty. It's things that we believe in collaboration, and we don't know it all. We know we cannot answer the questions by ourselves. So that's how we started the collaborations.
Thanks, Socrates. So it's been a quiet group today. I don't
I
see
know.
anything in the chat or the Q&A. And I did just double check that that the chat is open. But does anybody else have any questions? We have a couple more minutes. Okay, so excellent. Okay, Michelle. Oh, hold on. Let me. Michelle Smith, you should be able
Hi.
to talk.
Hey, Michelle.
Hey, Socrates. I just wanted to weigh in and say it is really good to see your face. I super appreciate your presentation of the current arrangement of the Human Food Program. As many times as I tried wrapping my head around it, I couldn't get it, but now I at least think I've got it. Um, and finally, the thing that really stands out that I want to personally say thank you is your emphasis on collaboration. Um, Collaboration between different labs, between different agencies, between educators, extension, and industry. Your closing statement that FDA does not know it all, um, and working with others helps provide the biggest picture is spot on. And so it's great to hear that. So again, thanks for everything.
No, Michelle, thank you. You were one of my mentors at the agency. I was very unhappy that you decided to look for greener pastures, but yeah, no, all the work that you guys did from the agriculture practice guidance that you guys put together, and all the interactions that you always did, and to make sure that we don't lose sight of that. Everything we do is to reduce the burn of illnesses, but it's public health, and again, right, collaboration, collaboration, collaboration. Mhm.
Socrates, I have a not specific question, but more of it's sort of I guess if you had a magic wand
Yes.
and you could um change one thing. Uh, is that
I have a list. I
your
have a
list of
list.
magic wand things you do? I'm just sort of curious because one of the interesting things in listening to you, right? So, you've worked as a PSN.
Mhm,
You've now worked in looking at research for outbreaks and
mhm,
the science of where the contamination is happening, how's it spreading. Yeah, I've certainly seen you give the cyclospora presentation that you did for FDA, which I have
mhm,
my opinions about that, which we won't get into. But I mean, you've been in a lot of those different areas.
mhm,
So, do you have a thought about, collaborate, collaborate, collaborate? Not always easy
mhm, mhm,
because
mhm,
of difficulties of communication. And so
mhm,
I'm just sort of curious, and is there one thing you would say, sort of like, man, if we could do this, or if we thought about addressing this, or if we understood this better, you know what I mean? Is there one thing that you kind of roll over in your head? Um, I have things I roll over in my head when I'm walking or when I'm swimming or whatever. And I'm interested what yours would be given your breadth of experience in all these different areas.
um, wow.
Oh, and Donna wants you to quit sharing screen.
Oh, thank you, because I don't want to say thank you anymore.
Although I do love this diagram with the produce intertwined with the DNA.
Yeah, yeah. To me, it's just that one of the concerns that I have is that there's a lot of people out there that we don't know about, that has a lot of information and the willingness to work, and because for one reason or another we don't know about them, we never work with them. If there was a way of really going to the basics and identifying people who knows what we don't know and start pulling them, that would be ideal. Because the biggest problem that I always have is like, if I'm thinking about this, somebody else either already thought it or is also thinking it. and having that forum, Betsy, to actually talk about the basic questions, but open to all the players, that would be ideal. And funny, because what were the questions that they asked me when they were doing the Produce Udall Reagan Udall
Yeah.
Foundation? That was
Mhm.
one of the things that I did. That's what we need. We need a forum like that. Yeah.
Fantastic. Thank you. I know we're short on time, so I'm going to stop talking.
Okay.
Excellent. Thank you, Socrates. I don't see any other questions, and we are a minute after time. So thank you, Dr. Trujillo. I really appreciate that presentation and for answering those questions. I did want to share screen, or I am sharing screen. I mentioned this in the chat box, but here are some of the upcoming PSA webinars. The next English language educators call will be with Dr. Shirley McAuliffe on September 15th. She's still working on the title, but it's going to be looking at bacterial growth and crop characteristics. And then I also wanted to just share some of the dates for the upcoming PSA trainers and lead trainers annual update webinar. All of this information will be shared with you in future educators groups. So with that, recognizing that it is 3:01, I am going to close this webinar, but thank you all for your questions and for logging in today. Thank you so much. And thank you again, Dr. Trujillo, for that great presentation.
Thank you, everybody. Stay well. Bye.